welcome to the 5th webinar of the series on emerging and re-emerging viruses. And today we have Dr. Sreekumar from Rajiv Gandhi Center for Biotechnology to deliver the talk on emerging prospects in COVID-19 therapeutics treatment. Dr. Sreekumar has extensively worked on dengue and chikungunya and published several papers in peer-reviewed journals of international repute, and has for many students. And he's an expert committee member of Institute of Advanced Virology, as well as an adjunct faculty in the institute. Currently, he is supervising the ICMR recognized diagnostic facility, that is the RT-PCR-based diagnosis of COVID-19 at Rajiv Gandhi Center for Biotechnology. Started his career as a veterinary surgeon after completing MBSc in immunology from Indian Veterinary Research Institute. He has pursued a PhD in biotechnology from Rajiv Gandhi Center for Biotechnology, and he has been a visiting faculty in molecular biology and immunology at Johns Hopkins Boomerang School of Public Health, Baltimore. And on behalf of Kerala State Council for Science, Technology and Environment and Institute of Advanced Virology, I invite you to the lecture. Over to you, sir.
Good afternoon. It's a pleasure to join this program. And since we are all into this COVID, and most of us are aware of this COVID scenario and about the virology, I thought I will share some points on how we are developing drugs on these viruses and with a little bit background on the disease. And already most of us are aware about the many aspects of this viral disease, and many of these things which I am going to talk to you, I'm sure it will be an overlap which you have already heard. But if I am able to convey some new things to you and some updated information, I will be happy that my presentation has served the purpose. So let me start with the presentation. I hope you can see the first slide.
Yes, sir. Yes, sir.
Okay. So the title is Emerging Prospects in COVID-19 Therapeutics Development. And as you can see, a large coronavirus in the field, and all of us are trying to tie it down with using different measures. And also you can see some people with cameras focusing on how we are doing it. And when we look at the 21st century where we are now, even from the late 20th century and even in the beginning of the 20th century, 21st century, we have seen that a large number of emerging infections are coming into the picture, and we have to tackle especially the swine influenza, which has been a major pandemic in the last decade. And we could see emergence of Zika virus infection and local incidents of Nipah virus infection in our own state. And we have been going through all these emerging diseases in the past. And incidentally, so far we have been studying only viruses which are very prominent in the picture. And all our focus has been always on to diseases like HIV or maybe hepatitis B, hepatitis C, and all those prominent diseases. But slowly we came to a situation where we need to focus more on neglected diseases, which so-called neglected diseases. And most of these diseases were in the tropics. And we see that though they are originally confined to the tropics, they are moving towards the subtropics and becoming a global issue. So in the coming years, we may see more and more new infections coming into our midst. And so any viral infection which has slightest possibility to become a more aggressive infection with its transmission potential, we cannot neglect now. Now the world itself has become a kind of a global village, and from one place to other place, a person reaching in few hours time can lead to the spread of the disease. Even the current COVID pandemic, we have seen how it is systematically spreading from one place to one country to the other country and slowly gaining its grip. So, any disease, almost all of these viral infections, especially those are mosquito-borne diseases, tick-borne diseases, or those with the unknown vectors, and also most importantly, the respiratory viral infections. All of them need a particular an augmented attention from our side. So even for the current coronavirus SARS-CoV-2, we had a predecessor SARS-CoV-1, the Hong Kong SARS, and then later on MERS coronavirus. So after those infections subsided, especially the SARS-CoV-1 subsided with 8,000 cases and 800 deaths in almost 8 months' time. Maybe we stopped or maybe reduced the speed of the research in this field, and the investments also were not much in the research arena for the SARS. So the momentum has come down drastically, and now when we have a second variant of the SARS coming in, we don't have much to rely upon other than some of the basic techniques that we have devised, and we don't have a drug which was developed for the SARS that will be used for SARS-CoV-2, or we don't have a vaccine that might have been useful in this particular situation, also because these viruses are closely related. So, at any point of time, even though we don't see the continuation of an infection, we may have to do our bit of research into that, expecting that this may re-emerge at any time. Another good example would be chikungunya virus, which surfaced maybe some 35 years back and subsided later on, to come back into our into the world again maybe in 2002 during that time. So again, during the chikungunya outbreak, we had to see that this is a new. We have to treat it as a new infection, and also start again from scratch to build make vaccines, make drugs, and so on and so forth. So most of the time now it is giving us a picture that we have to always be prepared to tackle all the unknown viruses that is going to emerge, and also continue working on these diseases which has already emerged and subsided, thinking that it can re-emerge at any time point. Now, yesterday I was just looking at the current scenario of SARS in the world, and you could see around 89 lakhs of people are affected by the disease currently, and a huge number of deaths have been caused. Interestingly, those countries like United Kingdom, Spain, Italy, Germany, and Turkey, which were initially overburdened with the disease and they had their share of the maximum number of cases, and you can see that now it has come down, and they are back into the baseline, and probably most likely their peak is over and may not see a second bout of the disease. But what is happening in India? We are still in the exponential phase of the outbreak, and we don't know when is the peak is going to come, whether it is going to come next month or a few months later. But we already have surpassed the total number of cases which has already been happened in the other countries who had already seen the peak. So we have almost 440,000 cases currently, and even we regularly now see that the number of cases are increasing. Even in Kerala, if you see the number of more number of expectators are coming, and the actively diagnosed cases are increasing day by day. So from the 2-digit number, we for the last week continuously we are running into a 3-digit positive number of cases, and we are strongly suspecting. Initially, we were doubtful about the community transmission, but now more and more people are getting convinced that there is a community transmission, at least in the beginning stages, happening with many cases which cannot be traced to a contact. Now, as we all know, this COVID-19 caused by the SARS coronavirus 2 affects the primary site of infection is the lungs, where it is focused, or its infection happens in the type 2 pneumocytes, which can express the ACE2 receptor angiotensin converting to an enzyme to receptor, converting enzyme to receptor. And from the site of infection, it can disseminate to other organs because this ACE2 receptor has distribution in many other organs. But we also know that this ACE2 receptor expression alone is not sufficient for the virus to infect these cells, because the spike protein has to be primed by another serine protease called the GMPRSS. And the presence of these core receptors are also important for the virus infection to happen, effective infection to happen. So, but there are other organs which express these core receptors, especially the intestinal epithelium, and they are also, along with the ACE2 receptors, that can efficiently infect. And many times what happens is it leads to a condition called viral sepsis in very complicated cases with the multi-organ involvement, especially the heart, the kidneys, and also the liver. And a number of organs are affected because of the impact of this virus. Lungs, as I said, it is the primary organ that is getting infected. Subsequently, there can be infection in the heart, and it can lead to the blood clots, heart attacks, and cardiac inflammation. And in many cases, it can invade the brain, leading to maybe the patient will show some confusion. There can be strokes, seizures, and other inflammatory conditions to the brain. And the virus is also thought to cause conjunctivitis. Then it can infect the nasal cells, nasal epithelial cells, causing loss of smell, anosmia. It can infect the liver cells, leading to changes in the or causing damage in the liver. It can cause kidney failure. It can lead to infection in the intestines, and many people who have this initial stages of the SARS, they present with diarrhea-like symptoms. And as you can see, the death rate in the infection with the COVID among different age groups, if you see the people who are 80 plus, they have around 14% of probability of a death. And as we go down in the age group, like the kids who are less than 9 years of age, the death rate is almost nil. And depending on the age, there is a significant variation in the death rate. And it is presumed that those children who have, who are less than 10 years or 9 years, the amount or the level of ACE2 expression is comparatively low, so there is no effective infection. That is one of the presumed thinking about the why the younger children are not very seriously affected with the disease. And in the older people, coupled with the expression of the ACE2 receptor, there can be immune senescence and also the comorbidities, which is increasing that. On an average, the mortality rate comes around 7%, 6 to around 3 to 4% in the population. But there is a significant stratification in the death rate among the different age groups. The common symptoms of COVID-19 and the timelines, if you see, these are some of the common symptoms that all these symptoms may not be present in the same patients, but more than one or two symptoms can be present simultaneously. See, as you can see, the exposure to the virus, the usual incubation period is around 2 to 14 days. Compared to other coronavirus like SARS coronavirus, it is around 2 to 7 days. MERS, it is again 2 to 14 days. Swine flu and seasonal flu, which belongs to another class of virus, the incubation period is only 1 to 4 days. That is much faster. But on an average, we can say it is around within 5 to 6 days as the incubation period. So from the exposure, if you take -5 as the date of exposure, so after 5 days, the symptom starts. And the initial 5 days, are considered to be a very, stage 1 of the disease, where you may see these kind of symptoms like nausea, fever, headache, diarrhea, sore throat, then dry cough, loss of smell or taste, fatigue. So these are very generic symptoms, which is not very specific as to COVID-19 itself. There can be many other diseases, many other, especially respiratory diseases, which can present with these symptoms. But in general, the sore throat and the dry cough, especially in this season, may characterize a chance for presenting with a COVID. And fever, it need not be present in many cases. And the loss of smell and taste is also present only in a percentage of population. The shortness of breath usually starts after or become more pronounced after a 5th day of infection. And as the days progress, the progression of the disease is decided. And in mild cases, as we see now, in the mild COVID cases, the virus shedding or the infectiveness of the patient is only in the early phase up to some 8 days. And as you can see, it starts few days ahead of the onset of the symptoms. And this is one of the major reasons
why we see this infection silently spreading among the population. Because even before a person comes to know that he is infected or he is developing symptoms of COVID-19, he has already transmitted the disease to people who are in close contact. Theoretically, we can detect the virus even at the very early stages, but we will not know whether the person is exposed or affected. But usually, after all the onset of the symptoms, we can detect the virus in the nasal nasopharyngeal swab. The technique that we use is reverse transcription polymerase chain reaction.
Since this is an RNA virus,
we have to reverse transcribe the RNA. Supplementary DNA, which is amplified, and this is a real-time PCR system that we use for detecting the virus. And the virus detection can be even up to some 15 days we can detect the virus. But actually, even though we are able to detect the virus up to 15 days, the virus which we detect at later time points, like some 9 days, 10 days, like that, it will not be in the infectious part. It will not be an infectious part. It will be
inert viral particles, and only the nucleic acid will be there. So that is why we see that the virus shedding or the infectiousness of the patient is only up to around 8 days in the case of a mild COVID infection. And we can also see that the inflammatory phase is around 8 to 10 days. The peak
around 8 or maybe a week after the onset of the symptom, the inflammatory phase or the peak symptom starts in a mild case, and the patient recovers in about 2 weeks' time. And even just before the symptom relief, it's also we may be able to detect some amount of the in the patient by RT-PCR, but the patient as such is not infectious to others. But in the case of severe COVID, the symptom onset, when the pre-symptomatic transmission happens definitely, the virus shedding can continue a little more, maybe 10 to 12 days, the virus shedding can continue. And also, the detection of the virus is also a little more prolonged. We can detect so we have from Kerala, we have cases even where we detect the presence of the virus around the 35 or 36 days post recovery or post onset of symptoms, I would say. Then the hyper-inflammatory phase, where along with the complication, it starts little later, maybe by 14 to 15 days it peaks. And this patient will be in a more complicated stage of the infection. So in the initial stages, as I said earlier, it will be fever, dry cough, diarrhea, headache, loss of sense of smell or taste, possible thermal, neurological or cardiac disorders. Slowly the shortness of breath and hypoxia starts and leads to acute respiratory distress syndrome or inflammatory syndromes and shock, cardiac failure, and in some cases there can be coagulation disorders, and and slowly the disease progressed from the stage 1 to stage 2 to stage 3. And as we see, when we are looking for antiviral drugs or therapies, not only antiviral drugs, other modes of therapies which can be used to save these patients from complications. Now, what are the currently used treatment or clinical management approaches for COVID-19? One of the major approaches is non-pharmacological treatment. That includes the supportive and symptomatic care. So essentially, it starts with bed rest, adequate nutrition, water and electrolyte balance. Sorry, there is some problem with my presentation. I'll come back in a minute.
Okay, sir, no problem.
Okay, are you able to see now?
Yes, sir. Yes, sir.
Okay, so essentially in the mild cases, this bed rest, adequate, all these initial approaches should be fine. Especially if there is fever, we can use antibiotics like paracetamol, not other non-steroidal anti-inflammatory drugs, and cooling measures. So these are recommended. And as the condition prolongs, there needs to be an intensive monitoring of vital signs like blood pressure, respiratory rate, heart rate, and most importantly, the oxygen saturation. So this is one of the major reason this condition called happy hypoxia. This is one of the major reasons for sudden death that is seen in COVID patients, especially those who are in the younger ages or maybe do not have comorbidities. And this happy hypoxia is a condition wherein the saturation level, oxygen saturation level, see the cutoff saturation is around 93% of SpO2, and when even in these patients who develop this happy hypoxia, they will not be feeling or they will not be dyspneic, they will not be having any difficulty in respiration, even when their blood oxygen level goes much below this 93%, even up to some 70% or 60%, they may not develop any difficulty in breathing, or they will not feel that they don't have adequate oxygenation. And suddenly they collapse and it can lead to mortality. So this is a symptom which is a silent killer in many COVID patients. So even in those patients who have these mild symptoms and who are advised to stay home and look for any complications or any further exacerbation of the symptoms, this oxygen saturation has to be monitored properly, and it can be done very easily with a pulse oximeter, which is a very small handheld device, and it does not cost much. It's around 3,000 to 4,000 rupees, which can be just applied on the tip of the fingers, and it can readily give you the oxygenation level. So, and also monitoring laboratory markers of disease progression and clinical outcomes are also important. And this needs to be done as per the doctor's discretion if they want to look into the prognostic markers, especially those that can describe the severity of the infection or give some idea whether this patient can go into a more severe condition. And in more complicated cases of COVID, the patients, when the blood oxygen saturation level falls less than 93%, these oxygen supplemental oxygen treatment has to be initiated. So it can be with simple mask, oxygen supplementation using simple mask, and which is done regularly, or high flow nasal cannula based oxygen therapy, non-invasive positive pressure ventilation, invasive mechanical ventilation using ventilators, or extracorporeal membrane oxygenation. So these are the different approaches that would be used in an emergency care facility for improving the oxygenation level in the patients. And also, while treating these patients, involvement of other organs has to be monitored. See, this happy hypoxia and early oxygen saturation monitoring is very important to avoid secondary damage to other organs. So we need to always see that the oxygenation level is always maintained above this cutoff limit. And also, the treating clinician will look into the comorbidities of the patient, and accordingly, the patient will be treated. And also, most of the time now, we ourselves are hearing or listening to all the cases of COVID, and we are getting more anxious about this condition. So, in all those patients who are diagnosed positive, they will be very anxious about their outcome, and also, so we need anxiety management measures and also psychological support to all the patients who are diagnosed positive in COVID diagnosis. Now, the pharmacological agents, which we will come back again little more detail, are the most commonly used are the antiviral treatment. Specific antiviral treatment is not available as such for COVID or SARS Cov-2. But these are the general drugs that have been, especially most of these drugs which I have taken here is from a publication that has consolidated all the information that was used in China. What we are doing currently in India is it is yet to be published, but more or less it is as per the WHO guidelines and other experience from other countries, and it will be more or less in the similar lines. So interferon alpha 2 is one drug that has been used either as mostly as an inhalation form. Lopinavir or ritonavir has been used as a combination. Ribavirin has been tried. Chloroquine has been tried. Arbidol is another drug which was used, and remdesivir is now being used in a wider way to treat as an antiviral measure for COVID-19. Glucocorticoid therapy as a supporting therapy, especially many studies say that it works more. Maybe in mild cases they don't prefer to use glucocorticoid therapy, but in advanced cases they try to use this methylprednisolone. Or now recently there have been much hype on the use of dexamethasone as adjunct treatment in clinical management of COVID. Convalescent plasma, especially plasma from patients who have been recovered, and those plasma which is collected from the recovered patient by a method of apheresis, so that all the RBCs from the patient will be returned back to the patient, only the plasma will be collected. And that patient who is subjected to apheresis, they can donate the plasma more frequently than the routine blood donation. So those patients' plasma is subjected to ideally it should be titrated for the presence of anti-COVID antibodies. And there is a cutoff like 1:320 or 1:640. The minimum cutoff is there for the viral titer or the antibody titer. And those convalescent serum is administered to the patient to neutralize the virus and reduce the side effects of the viral infection or control the virus infection. Major complications, biological modulators, especially the cytokine, is one of the major reasons for complication in COVID is the cytokine storm. And to block the cytokine storm, especially the effect of the IL-1 or the IL-6 cytokines, there are some molecules available. One is IL-1 receptor blocker called anakinra, and there is another one called the tocilizumab, which is an IL-6 blocker. This is also put to use in managing the COVID-19 patients. Now coming into a little bit about the coronaviruses, which is the virus in SARS coronavirus. So this is the order Nidovidae. So there is the Coronaviridae family, Orthocoronavirinae subfamily. There are four genuses. There is alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. And among that, this alpha coronavirus, they are human coronaviruses which can cause upper respiratory tract infections. And there are in the beta coronaviruses, we have the other human coronavirus HCO-43 and HKU1, then SARS coronavirus 2, and MERS coronavirus. Then we have the avian coronavirus in the gamma coronavirus. And this is an artist's vision about the coronavirus and all the major proteins which are this nucleocapsid protein, the viral RNA, the envelope protein, and membrane glycoprotein, the spike glycoprotein, all those are depicted in this. And these spike proteins which are projecting outside, this is a major protein which is involved in the infection process binding to the receptor. This is the spike protein out that will bind to the human KSCSTE receptor along with the presence of co-receptors. This will be helping the virus to enter into the system. This human coronavirus, which has a long history, if you see even the coronaviruses that cause upper respiratory tract infections, they have been identified long time back. The presence has been dated to long time back. And the recent emergence is the SARS Cov-2, which happened sometime in 2003. Then the MERS coronavirus, and subsequently, now recently we have the SARS Cov-2. Now this SARS-CoV, on the right hand side you can see the genetic organization. It is almost the same for almost all these coronaviruses, including the bat coronavirus, camel coronavirus, all these mouse MHV, and all these viruses have a general scheme or a more or less common genetic organization. And this is the genetic organization of SARS-CoV coronavirus, which is around 30,000 kilobases, which has a long OR of 1, which can code around 7,000 amino acid long protein. And there is an 4,382 amino acid protein. And there is an OR of 1B, which will code for 7,073 base pair protein. And all these are non-structural proteins which are important in assisting the virus to replicate, especially the proteases like the 3CL protease. And then there is RNA-dependent RNA polymerase. And all these are important in the viral replication process. And there are structural proteins expressed by the 3' region of the genome. And interestingly, this virus also has a poly-A tail. It's a plus strand RNA genome and mimic the host mRNA with a 5' cap and a poly-A tail. So, injecting this RNA alone, this virus can replicate. All these positive stranded RNA can replicate and produce progeny variants. And this structural protein form the structure of the virus or the shell of the virus, and in that the spike protein is the most important one that is mediating the infection process. So this organization, I'll come back to the slide once again, this is very important because we for developing drugs, we would like to target many of these proteins, especially the 3CL protease, RNA-dependent polymorphism. These are some of the common proteases, common targets which is used in targeting these viruses. Now, coronaviruses, the cellular receptors. So, for all these coronaviruses, there is a natural host. Some have a natural host as bats. Some have got this rats as a natural host. And most of them, as you can see, the bats are a common source for coronavirus. And usually, from the bat, as you can see from here, even though this bat is a natural host, they need an intermediate mammalian host before it jumps to human being. So that is why even we see so much of bats around in our country or in many places, even in the forest. It is very unusual that there is a direct jump of this organism, any viral organism from the bats to human population. Even in the case of Nipah virus, we presume that it would have jumped from the bat to human, but definitely there would have been an intermediary host, a mammalian host, which we would have missed to find out. But in all the known cases, there is a mammalian host, and it can be any animal, in case of the MERS coronavirus, it is camel. In case of SARS coronavirus, it's a there are different mammals which are included, involved in this process, and you can see a number of receptors used by these viruses, which are specific to each category of this virus. The ACE2 receptor, and some viruses use the aminopeptidase receptor, and some viruses use the acetylated salic acid, or acetylated salic acid residues as the receptor. And in case of MERS coronavirus, it is the dipeptidase peptidase 4 as the receptor. And we also see that these receptors are expressed in many tissues, not only in the lungs, but it can be present in many other tissues. But as I said earlier, the efficiency of infection into all these tissues is not as good as in lungs or maybe in other cells, like which also express a co-receptor, like called as the protease that is required for priming the spike protein, that is the GMPRSS2. So the secretory goblet cells in the nasal mucosa, or the type 2 pneumocytes in the lungs or the absorption and in the small intestine, they are very efficiently infected or proactively infected by the COVID-19 virus. And when you look at the phylogenetic relationship of this, you can clearly see how these lineages are clustering. And each one, this alpha coronavirus, beta coronavirus has got three lineages, lineage A, B, and C. And the SARS Cov virus belongs to the beta coronavirus lineage B. And these are the pangolin, Chinese pangolin, or the bats are considered or thought to be as the sources of this SARS Cov-2. And within the SARS-CoV-2 strains, we see a lot of genetic diversity, especially with large number of cases coming up. These RNA viruses, since the RNA-dependent RNA polymerase have over fidelity, each replication cycle, you will see more number of mutations accumulating, and after some time, some of these mutations get stabilized, and that will even change the properties of the virus. And we have done a small analysis of the published SARS-CoV-2 genome, which includes genomes from around 50 strains from India. So we sampled around 5,000 genomes, which was available at that time in the GenBank. Now it has almost crossed 20,000 genomes in the GenBank. So from that we sampled with 289 strains and made a phylogenetic analysis. And you can see distinct clustering patterns or clade patterns in this. And you can see that how this lineage is evolving. And you can see this virus from India, which is marked in red. They belong to different distinct clusters. So we still don't know whether these clustering differences have got any phenotypic effect in the virus, especially with respect to the infectivity, the severity of disease that is seen in the patients, and whether these differences. We have done some analysis of the spike proteins and tried to predict how this affects the ACE2. And we could see that some of the mutations definitely increased the affinity of the ACE2 receptor binding, indicating that this virus may be very strongly strong affinity to the ACE2 expressing cells, and the infection may be better. But there are mutations which also reduce the affinity of the spike protein to the ACE2 receptor. So, unless we are able to connect it with the clinical outcome in the patient, which is not available in the database, we could not do that at that time. It's always anyone's wish to connect this and make this kind of an information, because that will tell us more about how this virus is evolving and whether there is any severity changes, whether the kind of an attenuation or the reduction in the virulence which we hope that will happen in due course, or unfortunately an increase in the virulence of the virus because of some critical mutations enhancing its properties. So, which we don't wish to happen. But those kind of analysis can be done only when we have a clinical correlation and the viral sequence correlation when made possible. Now, the infection cycle for most of these viruses, including the SARS Cov-2, for the positive sense RNA viruses, and it is a receptor-mediated endocytosis, and the virus gets into an endosome. And inside the endosome, there is a major change that is happening once that endocytosis happens and endosome is formed. This endosome compartment is acidified, during which time these viral proteins, especially the envelope proteins and the spike proteins, they all undergo conformational change, and this envelope, along with this envelope protein, facilitate the fusion of this envelope membrane with the endosomal membrane. And this will open up the channels and will help the nucleic acid, the viral genome, to be delivered to the cytoplasm. Since this is an mRNA or it's a messenger sense genome with all the properties of a messenger RNA, it will immediately into viral proteins. And these viral proteins at the same time helping in making more and more copies of the mRNA, which will also help in making the structural proteins, which will help in assembling or packaging the replicated RNA, and finally everything gets assembled on the membrane along with the spike protein incorporated into the membrane. This again forms in the vesicles, which is getting released out as a new progeny virions, and this process continues. The research priorities, if you look at in SARS Cov-2, you can see the research priorities in 4 major domains. One is in the diagnostics, which we are seeing a large number of people or companies and research institutions are involved in developing more or better diagnostics or faster diagnostics for SARS Cov-2. So it can be either by detecting the virus itself, or it can be by looking at the immune response against the viruses. And some of these diagnoses can be for looking at the control of the infection, how the separation is progressing. Now, there is an augmented research going on in candidate vaccines. And so we need to, when we go for a vaccine development approach, we have to have multiple parameters addressed, like how to check the efficacy of the vaccine, how an assay can be developed to evaluate these vaccine response, then the target product profile. All these parameters people are trying to standardize and developing candidate. And there is a vaccine from most of these vaccines, especially that has reached to a phase 2 clinical trial, is an mRNA-based vaccine. And this vaccine, we hope that this will come out sooner to be used in the patients. Then genomics is a component of the basic research. Then monoclonal antibodies or viral understanding the viral pathways. All these comes part of it. And again, a lot of this investment is being done in identifying candidate therapeutics for the virus. Immune therapies, currently we are trying some of the immune therapies which is already available and or maybe used in other viral infections. Novel compounds are being screened, and also repurposing of the existing sorry, there is some problem. Okay, repurposing of the existing drugs is also being attempted. Now, drug therapy against viral infection can take 2 approaches. One is the approach which focuses on the virus, and second is an approach which focuses on the host. And in the approach which focus on the virus, our aim will be to increase, to inhibit the increase in the viral load and the spread. So this is targeting. This is done by targeting specific viral proteins or steps in the viral replication. That is very or it's particularly unique to the virus. It does not involve any of the host components. So there is no side effect to the host because we are focusing only on the viral components, which is not shared by host. But the problem is the viruses are very smart. So if we keep on focusing on this viral proteins, viral replications or entry, so they will mutate and they will develop resistance much faster. Another approach would be to focus on the host. The aim is to inhibit the cellular pathways that is hijacked by the virus from the host for its replication or survival. So while studying the virus replication in case of many of these viruses, we will be able to identify some unique cellular processes or pathways which is required for the replication of the virus and which may not be that essential for the survival of the host even if we stop it for some time. This will not affect the survival of the host cell, but it will definitely cut down the productive replication of the virus. So in this case, we are focusing on the host components, and so the virus will not be able to develop, or the chance of development of resistance by the virus will be much lesser. Definitely there can be, viruses are much smarter. Even in this case, also virus will be able to develop a resistance or maybe develop alternate mechanisms to overcome this. But the chances are comparatively much less. And but the problem is, since we are targeting some of the host pathways, there is a possible chance of side effects to the host system. So, but these two approaches, when we combine them judiciously, we'll be able to get a much better outcome than using either of these approaches. Okay. Now, the antiviral development, when we focus on the virus, we just, as I said earlier, for the COVID coronavirus, the viral entry happens with fusion, uncoating, and these are different steps: the genome replication, the assembly, release. So, any of these steps we can target to develop an antiviral once we have very good understanding what are the mechanisms behind it. So the common approach would be to use an entry inhibitor. So it can be any molecule that will either bind to or prevent the virus from binding the receptor, or maybe preventing the fusion by different mechanisms, or at the time of the release of the virus. For example, we hear about oseltamivir or Tamiflu, which is an inhibitor that prevents the release of the influenza virus from after the replication. So at any of these stages, we can use this entry inhibitors, or we can focus on the replication inhibitor, which will be focusing on some of the key enzymes or proteins that is involved in the replication of assembly of the virus. So in the COVID coronavirus, we have both these approaches, as I said earlier. So the blue colored ones are targeting a virus, that is the spike protein, the RNA-dependent RNA polymerase. Then the viral proteases, especially the 3-CL protease, is also known as the main protease, or the PL protease. So here are some of the molecules that is already being used in case of COVID-19, which is targeting. For example, this is lopinavir, ritonavir, which is widely used to treat COVID patients, is a 3-CL protease inhibitor. It is not developed specifically for this SARS Cov-2, but developed for other viruses which has the similar kind of protease, and this was based on initial analysis. It was found that it might be useful for the SARS Cov-2 also, and that is being used. Since it's already approved for clinical use, it is being repurposed in case of SARS Cov-2. Many clinical trials are going on how efficiently we can use this. Another molecule, which is the RNA-dependent RNA polymerase inhibitor, is the remdesivir and favipiravir. And ribavir has different actions, including RNA-dependent RNA polymerase action, but it was not found very effective in many cases. But remdesivir is being now promoted as one of the possible drug for treating the COVID patients. Now, when I come back to the other molecules which are been mentioned in this picture later on. So, this is the approach where we focus on the virus, on the S protein, RDRP, and viral proteases. Now, in the case of host targeting, we can target the endosome, immune modulation, host proteases, and host cell receptors. Now, in this category, like where we look at the antiviral targeting the viral life, there are a number of attempts being done to repurpose the old drugs which are used in other diseases. Some of the examples, like for example, doxycycline is widely used and is a widely used antibiotic, and is being used as a prophylactic for in many times in leptospira illness to prevent especially the sanitation workers and all that from getting infected. So this has been found to have some antiviral effect, especially by upregulation. These are some of the perceived mechanisms by which this anti-inflammatory and antiviral effect of doxycycline happens. It upregulates a protein called zinc finger antiviral protein. It chelates zinc compounds on matrix metalloproteinases, which is important for the spread of the viral infection from one cell, along with the receptors. Then inhibition of the viral proteases, so it is thought that it will inhibit the papain-like protease of the SARS. It is also thought to reduce the pro-inflammatory cytokine. So, doxycycline is being projected as a very good drug for treating this COVID, and clinical trials are being done in many places for using this doxycycline in treating COVID patients. And this is a paper where they have published screening a large number of already known drugs against this SARS Cov-2. And this is a cytokinetic effect reduction assay. You can see those drugs which prevent the infection will retain this color, and those where this infection is happening, there will be clearing of this infection. You can see that some of these drugs, like gemcitabine, is able to inhibit the cytokinetic effect caused by the virus without much cytotoxicity. And this is another GS441524 is also able to inhibit. See, at these concentrations, there is no, especially 543 and all these concentrations, there is no replication of the virus happening. So these stained cells are intact here. Here you can see the clearing. So completely the cell has died, and then there is no staining happening, and there is no cytotoxicity for these drugs. So there have been a lot of efforts happening in screening or repurposing the old drugs against SARS-CoV-2. And another therapeutic approach that is being used for COVID-19 is focusing on the host immunomodulation. See, some of the studies have shown that when you look at the cytokine profile in COVID-19 patients, it overlaps with what we see in cytokine release syndrome, like this is the green circle, or immune reconstitution inflammatory syndrome, and that is this orange circle, and secondary hemophagocytic lymphohistiocytosis. So you can see there is a large number of key cytokine molecules, especially that IL-1, IL-6, C-reactive protein.
All these are getting overlapped in all these conditions. So us to think about why can't we use some of these cytokine storm inhibitors or anti-IL-1 and IL-6 molecules for treating this. And in many cases, this IL-6 inhibitor like tocilizumab is being effectively used in many patients. But there is no consistency in the treatment response. That could be one reason why this is not considered as a very specific treatment, but it's an adjunct therapy to many other treatments. Another molecule that is for treatment of the cytokine storm is being developed is called the ulinastatin. and it's a serine protease inhibitor with anti-inflammatory properties, including inhibition. This is also in clinical trial. And a number of cell-based therapies
are also being
used,
used,
like immunotherapy using NK cell therapy, T regulatory cell targeted therapy. These are all many times in the research phase and not into the clinics now. But some of them, like convalescent plasma therapy, is being currently used. Then neutralizing monoclonal therapy is a proposed treatment that is emerging. Then IL-6 and IL-1 inhibitors are already available. So a number of immunomodulatory approaches or immunotherapy approaches are being used, and many are being under development. And interestingly, as we look into the literature, we see a large number of natural products are being projected as antiviral agents and against SARS Cov-2. Essentially it is done using in silico approaches, and as few studies are appearing using in vitro approaches. And these are some of the references I could pick from the literature, where most of these studies are from India, where they are looking into the natural products, especially plant-derived natural products, into in silico studies. And recently, from ISERC Trivandrum, there is a study which has appeared where they could see that green tea and spirulina extracts inhibit three SARS viruses, coronaviruses, SARS, MERS, and SARS-2 spike pseudotyped virus entry in vitro. So this is an in vitro study which they have published as a preprint maybe a few days back. So there are a number of research approaches. This is all in the research side. Now, when you also look into the literature, you can see a number of dietary supplements and natural products.
There are some prior studies among other viruses or even in other infections, with other infections like human rhinoviruses or other respiratory SARS coronavirus, the MERS coronavirus. So they think that these supplements, especially the vitamin D as an immunomodulator, is being much projected, and many other natural products and dietary supplements are also being promoted as adjunct therapies for COVID-19. And a number of molecules are being under clinical trial, and some of them are in very advanced. Even there is one drug, umifenober, which is in the phase 4 clinical trial, and there are drugs which are in the phase 3 clinical trial. So it's a significant amount of trials are being done, and work is being done using already known drugs to be in COVID-19 treatment. Along with that, a number of monoclonal antibodies are also being tried out, especially as immunomodulators and for blocking the cytokines. And you can see some of them have reached up to phase 3 clinical trial. So once they cross these clinical trials, they will be available as licensed product in the market for treating patients. So already this anakinra and this tocilizumab is being used. Even it is available in India. It is being used in many Indian patients. Interestingly, in COVID-19 treatment, even stem cell therapy is also being used. Especially to protect the organ failure. So it's an advanced kind of approach, which is there are some few clinical trials for stem cell therapy. So even that aspect also is getting developed in a much significant way, whether we can address the organ failure with the stem cell therapy, or even inhibit cytokine storm with the stem cells. So this is also a new turn or twist in the kind of research that is happening in treating the COVID-19. Now, as I was telling, this focus on the virus, and also there is another approach, the focus on the host. So we see a number of compounds or molecules, drug molecule that is being used in targeting the host. And one of the major or very widely used one is the chloroquine and hydrochloroquine, which is targeting the host mechanism of the endosomal acidification, so that this acidification is prevented and this virus, there is no conformational changes in the viral protein and there is not able to enter in the cell. Then also the receptors, some of the host receptors targeting EMPR, serine protease. So there are some molecules which can prevent the function of this protease, so that the virus is unable to enter. And arbidol is also another entry inhibitor, which will prevent the fusion happening. And recombinant ACE2 receptor, human ACE2 receptor, if you use this will compete with the virus for this binding to the ACE2 receptor, and this will also prevent this virus entry. Then, as I said, the tocilizumab is an immunomodulator which will bind to the IL-6 receptor. So, along with the host virus targeted approach, there are a number of molecules that we can use in the host targeted category. Now, the chloroquine has not only the endosomal acidification, prevention of endosomal acidification, it also has multiple functions. So, increasing endosomal pH or inhibiting membrane fusion, host virulence, immunomodulation and anti-inflammatory action, glycosylation of ACE2 receptor and altered binding of this protein. So this might be unique with respect to the coronavirus infection. This chloroquine is also used in many other virus infections also, but where this ACE2 receptor is not very important. But here, in case of coronavirus, that would be one reason why there was much hype about this chloroquine and hydroxychloroquine use. In Indian ICMR has already has not advised against the use of chloroquine, but many developed countries, they are trying to stop the use of chloroquine because they could not find much use in the clinical trials. And also, there were some side effects for using this chloroquine, especially in cardiac patients in the QT interval changes. So this is not very in the Western countries. A number of cell-based studies have come out where they have looked at the signaling pathways modulated by the SARS Cov-2 and identified some of them could be used as the targets for developing antivirals, like NF-kappa, BTNF alpha signaling pathway inhibitors or IL-6 JAK-STAT signaling pathway inhibitors. All these are being developed as the potential molecules, like fingolimod is already an approved compound. So this can modulate these sphingosine receptors, and they have identified that sphingosine receptors have got some role in the viral replication. There are a couple of antibodies along with this tocilizumab. There is another one called sarilumab. There are a number of antibodies which is also being developed that can target multiple proteins. So, especially the host factors, so that this virus or the effects of the viral replication, toxic effect of the viral replication can be minimized. So, these all will come out at some point as adjunct therapy to treat this COVID patient. Another interesting aspect is how to utilize this interferon system for developing antiviral molecules. As we know, once this viral RNA or viral double-stranded DNA or RNA goes inside, it is sensed by the intracellular cytoplasmic sensors of the innate immune response. And this leads to two pathways actually. One is the protection of the interferon, alpha beta or type 1 interferon, and also the stimulation of the NF-kappa response, which will be an inflammatory pathway response. And this interferon response leads to production of interferons, type 1 interferons, which goes out of the cell, bind to the interferon receptor in the same cell or maybe in the adjacent cell, and leads to the stimulation of the production of interferon stimulated genes. Around 300 odd genes are present as interferon stimulated genes. And these genes, many of them have got direct antiviral effects, or some of them in association with other molecules prevent, they are actually the effectors of antiviral response. So interferon directly do not kill any virus or prevent replication, but they go and stimulate the production of interferon stimulated genes and produce the antiviral effect. So if we can tap this system, how to modulate this interferon stimulated gene expression, that could be one of the novel approach to develop antivirals. And one of our interests currently is how to tap this system in developing interferon stimulated genes and how to modulate them. Currently we don't have any molecule that is available which will modulate the production of this interferon stimulated genes directly other than using interferon. Interferon has a lot of other side effects, unwanted effects. But if we have any specific modulators of these specific interferon stimulating genes, that will become a very promising antiviral molecule. So one advantage is many of these interferon stimulated genes are broad spectrum genes, and as I said in the beginning of my presentation, we are looking forward to large number of new viruses which are coming out, and it will be very difficult for us if we focus only on specific viruses and specific antiviral molecules as a combination, rather than we need to focus on broad spectrum antivirals. And I consider that some of these interferon-stimulated genes would be one of the good targets to develop broad spectrum antivirals, which may be useful even in case a new virus infection emerges in future, as long as it uses the same interferon stimulated gene, or we can use the same interferon stimulated gene modulation to inhibit the replication of the virus. So, developing broad spectrum antivirals will be one of our major focus in the coming years, rather than focusing specifically. Definitely, both that would be required, but a combination of these two would be the most effective way to tackle the emerging infections in the future. So at present, there is no specific antiviral drug against the SARS coronavirus 2. The global efforts focusing both on virus as well as host targets are yielding very encouragingly, and many new molecules or combinations are in advanced clinical trials, pointing at a promising future in the combat against COVID-19. Thank you.
Thank you so much, sir. It was a very informative lecture. We have got a picture of starting from the biology of the virus to the life cycle, the various, the infection that is happening with this virus, and then the immunological status, immunological responses, and finally to various, therapeutic prospects. And we have got a couple of questions. I now request Arjun Prasad, scientist of KCST, to take up some of the questions.
Thank you, Sherry. Thank you, Sreekumar sir, for the wonderful presentation. Actually, it was really informative.
Other participants, please mute your microphone.
So the first question is regarding the severity. It's about the severity of the symptoms depend only on immune status or comorbidities, or is there a chance of genetic variation of the virus in the same population contributing to it?
Severity of the infection depends on all these three. It depends on immune status, it depends on the comorbidity, it can depend on the virus also. But In case of coronavirus that is circulating in Kerala, if you focus, or in India, if you focus, we don't see any difference in the severity as such. Even the perceived severity or the number of mortality happening across in many parts of the country, it's a reflection of the total number of cases. If the number of cases go up, naturally there will be we will see more number of deaths, and we will perceive that the virus that is circulating in those parts are more severe. But when you look at the genetic makeup of this virus, they are all, even though you see minor clade differences, and so far no one could see that it is strongly associated with any severe outcome. So whatever cases of death that we are seeing, when you analyze very closely, you see a case of comorbidity in many of them. And even after very aggressive clinical management, the doctors are unable to save them, and because it could be because of the overreaction that is happening in the form of a cytokine storm, or maybe immune senescence because of old age, and many of these people are unable to respond well to the medication. So there are these are all multifactorial. So all these three will contribute to the severity of this. But right now, the COVID coronavirus, we cannot perceive that it is mutated to a virulent form or a non-virulent form. That studies are still in the very beginning, so we will not be able to comment on that.
Okay, thank you, sir. The second question is, like, does microvascular thrombosis develop only in severe cases, or is it a general manifestation?
It is not a general manifestation, so it happens only in severe cases, usually with cardiac affections, and also when there is an extensive viral dissemination. So usually even this microvascular thrombosis happens when there is high viremia, and there is spillage of this virus into the bloodstream and getting into other organs in a larger number.
Okay. The next question is regarding the nanosponges. The recently reported nanosponges, can it be used as early inhibitors of the virus?
Yeah, can you repeat what is that?
It's regarding the nanosponges. The recently reported nanosponges, in which the question is whether it can be used as entry inhibitors of the virus.
I don't have an idea about these nanosponges. I'm hearing it for the first time. So I will not be able to make any reply on that.
The next question is regarding the immunological memory. How long does the immunological memory for COVID-19 last in human beings?
In case of SARS Cov-1, that is the Hong Kong SARS, there was a study which was telling that the IgG response can be detected even after 2 to 3 years. IgG response is one of the parameters that we can look for the memory. And it is only an antibody response, but the T cell memory we don't know. I could not get into any article which describes that. That is what I told is about the SARS-CoV-1. Now, coming to this COVID-19, we are still not clear how long this memory response is there. But there is one study which says that in some patients, there is a Nature paper which was telling that after 3 or 4 months, we are unable to detect any antibody, or the antibody levels are much low, especially the IgG antibody levels are much low in the patients. But they also conclude that even though the antibody levels are low, the cell-mediated memory cells that is present will be able to protect these patients from a second infection. Whenever we have sufficient memory cells, even a small trigger with a second round of infection will boost up the immune memory, and it will produce the effectors like antibodies and CD8 cells much faster. So that will protect the patient from getting infected. So right now, one study which is available says that it is around 3 to 4 months. You see there is a reduction in the IgG level. But whether it is true for all the people across the population and all that, we are not sure, because this is a new disease. So altogether, we are just passing through a few months of large-scale infection. So we may have to wait a little more to say very, elaborately on that. But definitely, they say that whatever immune response is coming in infected patients, that is sufficient to protect them from a second infection for a pretty long time.
Okay. The next question is regarding the status of development of therapeutics in Kerala or in India. What is the status?
I would say it is in very early stages, and most of the institutes, research centers in India, they are gearing up for establishing facilities for doing this kind of research. There are only less than 10 institutions who have this BSL-3 facility for handling the virus cultures. But there are some other methods like developing pseudovirions based assays, which can be done in a BSL-2 facility. So a large number of institutions are trying to do that, including us. And once we have the pseudovirion system ready, we will be able to screen molecules. And but that is only the very beginning, because it has to we have to do the in vitro development, then subsequently preclinical development in animal models, then phase 1, phase 2. So it will be a very long time before we come out with a real drug-like molecule for COVID-19. But right now, this is very much in the beginning stage.
Okay. The next question is regarding the infection rate in children. Like as ACE2 expression is less in infants, then also infection happens. So, what about the infection rate in children?
The infection rate in children, see, there are two things: whether infection happens, and the second is whether complications arise. In any cases, even if children get infected, they will have mild symptoms, and we don't see any complicated. So far, we have not come across a report, I think, from Kerala where children have got this complicated COVID-19 infection. So there could be mild symptoms in the children, and since this is a season of COVID-19, we will check them with PCR, and we will be able to detect the presence of virus, because the PCR will pick up even virus in very low quantities. So we will pick up the virus, but whether that clinical infection is been pronounced, or whether it is only a very mild infection, and whether there are any complications develop, that matters.
Okay. But still, I think as a kind of prevention, the government even has given guidelines to restrict the movement of children below the age of 10 years.
That is true. There could be two reasons. Since they may get infected even in a subclinical way, and when they come back, they may be able to infection to the elders in the house who will get a clinical disease, especially if they are older people, grandparents in the house, and they may become more susceptible to this infection. So that could also be one of the reasons why we prevent these children going out and getting this infection, or carrying this infection from public places back to them.
Next one is a little controversial question. Like there are a few number of claims that they are some have invented medicine, including Patanjali. So I mean, I think still we are unable to say an answer.
I
have an answer. I will say this is a medicine which is when being marketed by belief, not by science. Because no one has tested this medicine. I don't know specifically about Patanjali or anything, but most of these claims that is being made, as per my awareness, it is never being tested against the SARS coronavirus in any system. And even if we show some antiviral activity in an in vitro system, that will not tell us much. In an in vivo system, it may not work. There are hundreds and thousands of drugs which have failed in the clinic, preclinical, even in animal models, before it comes to the phase 1 event. So definitely, I will say that this is all being marketed just based on belief, not based on science.
Okay. But as as evident from the news reports, I mean, in countries like UK, Spain, and Italy, where the number of cases were so high in the initial phase, they are. I mean, it says the curve has flattened there. I mean, what might be the reason? They have achieved the herd immunity, or
See, it is not that even curve has flattened. It has come down back to baseline. That is what the graph shows.
okay, yeah.
That's a very interesting
Development, and even though they had to struggle in few months back with a large number of cases, now it's almost a very few number of cases which they are easily managing. And there can be multiple reasons for that. It need not be herd immunity alone. It could be because few of the studies which have done in the community there, they say the herd immunity is not that much. So there can be multiple reasons where the kit they have used is sufficiently sensitive to pick up all the antibody response and all that. That is another question. But apart from the herd immunity, there could be other reasons also. Maybe they have effectively done the social distancing or mask usage and prevent the transmission much effectively. Or just like what we were speculating in the beginning, whether there is any change in the circulating virus which has lost its potency to transmit into more number of people. That is also another speculation. Or maybe the government intervention or the functional intervention was much more effective in containing that infection by the social distancing measures or mask usage. Because other than that, we don't see any reason why this infection subsides. There could be only two reasons. One is the intervention by the man. Second is natural forces acting on the virus, attenuating it, or making its survival difficult. So we don't know unless when we do a very large scale analysis, we will not be able to make anything. But this is a fact. The total number has come down drastically.
So, what's your opinion? Can we see a similar situation in India?
Definitely, at some point, we will also see that this number of cases coming down. But I will not be able to comment what is that some point.
Yeah.
Because again, even between the states, we see a lot of difference. In Kerala, you see even now the number of cases are much lower than in other states. There could be two reasons. One is we are able to contain and put all the people who are coming from others, or almost all from other states or other places into quarantine. Second thing is maybe compared to other states, we are more observing this mask usage and social distancing in a better way. See, just like vaccine usage, we don't need that 100% of the people
use mask or
100% of the people should keep the distancing. Even if it is around 70% or 80%, that itself will give a kind of a social immunity among the people. So even in the road, if you spot one person who is not wearing mask or not wearing mask properly, it may not contribute much, but if too many people are doing it, definitely that will contribute. So even 70 to 80% of the people, if they are following the norms, maybe that is one reason why even when we consider that there is some social dissemination of this infection, it is not happening in a very large scale.
Okay.
So we will not be able to say when this will peak. As we intervene much, the peak will be delayed, and we will be able to reduce the number of cases also.
Okay. So another question is regarding if people are using drugs, especially to control BP and all, how it will have effect on how the patients respond to COVID-19.
It's a more of a clinical question which I will not be able to answer in a very foolproof way. Maybe a clinician will be able to, but so far the reports say these kind of medications don't affect the severity of the disease or even does not modify the disease. But I may be wrong because maybe it's a right question asked to a practicing clinician.
Okay. I think we have exceeded the time limit. Thank you very much, sir. On behalf of KSCST and all the participants here, I would like to express our sincere gratitude to Dr. Sreekumar for giving us such a great presentation and busy, very busy schedule. Thank you very much, sir.
Thank you for this opportunity, and it was very nice speaking to you. Yeah. And I also see that a large number of people have attended, and I thank all of them. I hope that I could do justice to your time. Thank you.
Thank you all participants for joining us in the webinar. Thank you very much.